Here's something truly remarkable. Every organism you see, no matter how complex, started as just one single cell. That includes you. Your life began when a sperm and egg fused into one tiny cell. Now you're made of trillions of cells. How does that happen? Through cell division. And that's what we're studying today. All cells reproduce by dividing into two, with each parent cell giving rise to two daughter cells. These daughter cells can themselves grow and divide, and the process continues. This cycle of growth and division is what allows a single cell to form a structure consisting of millions of cells. We call this entire process the cell cycle. But here's the thing. These processes don't just randomly happen. They have to be perfectly coordinated. Three major things occur during the cell cycle. The cell grows, its DNA gets replicated, and eventually it divides. All three must occur in the right sequence at the right time. If they're not coordinated, you end up with daughter cells that have broken or incomplete genetic information. That would be catastrophic. The cell growth continues throughout the cycle, but DNA synthesis, the copying of genetic material, that only happens during a specific time window. And the remarkable part, these events are under genetic control. The cell has built-in checkpoints and regulatory systems to make sure everything happens in the proper order. Let's talk about time. A typical human cell divides roughly once every 24 hours. That's your baseline. But here's what's interesting. The actual cell division, the visible part where the cell physically splits into two, that only takes about 1 hour. Just 1 hour. That means the cell spends more than 23 hours preparing. In fact, if you do the math, the preparation phase takes up more than 95% of the entire cell cycle. Most of the time, the cell is getting ready, not actually dividing. And this timing varies wildly across organisms. Take yeast, for example. These single-celled organisms can zip through an entire cell cycle in just 90 minutes. They divide so much faster than human cells. This shows us that the cell cycle duration isn't fixed. It varies from organism to organism and even from cell type to cell type. The cell cycle is divided into two main phases. The first is called interphase. That's the preparation period. The second is M phase, where M stands for mitosis. M phase is when the actual cell division happens. The interphase is literally the phase between one M phase and the next M phase. It's the waiting, preparing, getting ready time. Think of it like the difference between training for a game and playing the game. The training is long. The game is brief. That's basically your cell cycle. Interphase is called the resting phase, but don't let that fool you. The cell is definitely not sleeping. It's incredibly active. During interphase, the cell undergoes both growth and DNA replication in an orderly, coordinated manner. To understand what's happening, we break interphase into three distinct subphases. These are G1 phase, S phase, and G2 phase. The G stands for gap, meaning the gap between DNA synthesis. Think of it as gap 1 synthesis, gap 2. Or, if you like, prepare, copy, and prepare again. G1 phase comes right after the cell finishes dividing. It's the period between when mitosis ends and when DNA copying begins. During G1, the cell is working hard. It's metabolically active, using energy, breaking down nutrients, building proteins. The cell is growing, getting bigger. But here's the key. It's not copying its DNA. That comes later. Most of the organelle duplication also happens during this phase. So the cell is making new mitochondria, new chloroplasts if it's a plant cell, new ribosomes, and other structures it'll need when it divides. Now we get to S phase, the synthesis phase. This is when DNA replication happens. The cell copies all of its genetic material. So if the cell starts with a DNA content we measure as 2C, by the end of S phase it has 4C. The DNA amount has doubled. Now here's something that might seem confusing at first, but it's really important to understand. Even though the DNA amount doubles, the chromosome number stays exactly the same. If your cell had 2 N chromosomes, that's the diploid number, before S phase, it still has 2 N chromosomes after S phase. The chromosome count doesn't change. What happened is each chromosome now consists of 2 copies, 2 sister chromatids, instead of just 1. In animal cells, while DNA is being replicated in the nucleus, something else is happening out in the cytoplasm. The centrioles are being duplicated too. This is important because these centrioles will help organize the spindle fibers during cell division. After S phase comes G2 phase. This is another growth phase. During G2, the cell is still growing, still getting bigger. But now it's also synthesizing proteins, specifically proteins needed for mitosis. It's like the cell is gathering all its tools for the job ahead. It's staging equipment, preparing machinery. By the end of G2, the cell is ready for M phase. It's fully prepared to divide. This is so important, I'm going to emphasize it again because it's crucial for understanding cell division. Before S phase starts, the cell has a certain amount of DNA. Let's call it 2C. Each chromosome has one DNA strand. During S phase, every single DNA molecule gets replicated. The cell now has twice as much DNA 4C. But, and this is critical, you still have the same number of chromosomes. That's because each chromosome now consists of two identical DNA strands, two sister chromatids held together at the centromere. After mitosis, when those sister chromatids separate and move to opposite poles, each daughter cell ends up with the same amount of DNA as the parent cell had originally. Each daughter cell gets 2C and each has 210 chromosomes, perfect genetic copies. So here's the DNA content timeline. In G1, the cell has 2C. During S phase, it jumps to 4C. It stays at 4C through G2 and into M phase. Once the sister chromatids separate during anaphase, you're creating new individual chromosomes with 1C each, and the DNA content of the cell space drops back to 2C. This is why comparing a somatic cell in the middle of M phase to a gamete can be tricky. If a somatic cell has just completed S phase and has 4C worth of DNA, and a gamete has 1C worth of DNA, then that somatic cell has 4 times more DNA than the gamete. This is an important comparison for understanding reproduction. Now, I want to make something clear. Not all cells are constantly dividing. Some cells in your body have basically stopped. Some cells don't divide at all after they mature. Take heart muscle cells. Your heart can't afford to stop working, so those cells stay put. They don't divide. Many other cells in your body only divide occasionally, when they're needed to replace cells that have died or been damaged. These cells exit the normal cell cycle and enter what we call the G0 phase, or quiescent stage. Here's what's interesting about G0 cells. They're not just sitting there doing nothing. They're still metabolically active. They're performing their normal functions. A liver cell in G0 is still processing toxins. A neuron is still transmitting signals. These cells just aren't dividing. They're not proliferating. But, and this is important, they can come back. If your liver needs to regenerate because of damage, those G0 cells can be called back into the cell cycle. They'll start dividing again. It's like they're in a flexible pause state, ready to go if needed, but happy to stay put otherwise. Your body decides based on what's needed. Now we get to the dramatic part. M phase is the most spectacular period of the cell cycle. During M phase, virtually every component of the cell reorganizes. The nucleus breaks down, chromosomes condense, the spindle apparatus forms, everything moves. It's orchestrated chaos, but it's controlled chaos. M phase is also called equational division because the chromosome number in the parent cell equals the chromosome number in each daughter cell. The genetic information is conserved. We divide M phase into two main parts, karyokinesis, which is nuclear division, and cytokinesis, which is cytoplasmic division. But remember what I said earlier, even though we divide these into separate stages, it's all one continuous process. You can't draw sharp lines between prophase and metaphase. One flows into the other. It's progressive. The science has been clear for a long time, but the evidence from this past decade is definitive. This period from 2015 to 2025 will be remembered as the decade the consequences of our inaction became undeniable. That temporary breach of 1.5 degrees served as a final, unambiguous warning. The scientific challenge now is to monitor these accelerating changes, but the human challenge is to finally close those gaps between promises and performance before those tipping points are crossed for good. Prophase is the first stage of mitosis. It comes right after S and G2 phases. During prophase, something major starts happening. The chromosomal material condenses. Remember how I mentioned that during S and G2, the DNA isn't organized? It's sort of tangled together. Well, now the cell starts the process of untangling all that DNA. It's compacting it, condensing it, organizing it into visible structures. The chromatin, that loosely organized DNA, becomes increasingly compact. The chromosomes start to become visible. As prophase continues, each chromosome is now clearly made up of two sister chromatids. These are identical copies, remember? They're attached together at a point called the centromere. At the same time, the two centrosomes that were duplicated back in S phase are now moving toward opposite ends of the cell, toward the poles. From each centrosome, long filaments of microtubules are radiating outward. These radiating fibers are called asters. They kind of look like little stars, which is where the name comes from. These asters are preparing for the big job ahead, pulling the chromosomes apart. The two asters, together with the spindle fibers connecting them, form what we call the mitotic apparatus. It looks like two stars on opposite sides of a cell connected by lines. That's the spindle apparatus. By the end of prophase, something dramatic has happened. Look through a microscope at a cell in prophase and you'll notice the nucleolus is gone. The nuclear envelope that was surrounding the nucleus, completely broken down. The Golgi complexes, not visible anymore. The endoplasmic reticulum, gone. The cytoplasm is getting crowded with chromosomes and the spindle apparatus. Everything else has disappeared or become invisible. The chromosomes are now the dominant structures you see. The cell has reorganized everything in preparation for division. Metaphase is stage 2. By metaphase, the nuclear envelope has completely fallen apart. The chromosomes are spread throughout the cytoplasm. The condensation of chromosomes is complete. They're as condensed as they're going to get. This is actually the best stage in the entire cell cycle for scientists to study chromosome structure and morphology because everything is so clearly visible and so organized. Each chromosome at metaphase is made up of two sister chromatids. These are the two identical copies joined together at a central point, the centromere. On the surface of the centromere, there are small disc-shaped structures called kinetochores. These are absolutely crucial. The kinetochores are the attachment points. This is where the spindle fibers connect to the chromosomes. Without kinetochores, the spindle fibers couldn't grab the chromosomes and move them. Now here's how metaphase works. All the chromosomes line up right at the equator of the cell, the middle. It's like they're all standing on an invisible line. This line is called the metaphase plate. How do they stay there? Each chromatid of each chromosome is connected via its kinetochore to spindle fibers, but here's the brilliant part. Each sister chromatid is connected to spindle fibers from opposite poles. One chromatid gets pulled from one direction. Its identical sister gets pulled from the opposite direction. They're perfectly balanced, held right in the middle. The cell is now perfectly set up for the next stage. Anaphase is where things really happen. It's the dramatic moment. At the onset of anaphase, every single centromere splits simultaneously, all across the cell at once. The sister chromatids that have been attached together, that have been held by spindle fibers being pulled from opposite directions, they suddenly separate. Now we call them daughter chromosomes because they're separate entities again. They're no longer pairs. And they start migrating toward opposite ends of the cell. Here's something really interesting about how the chromosomes move. The centromere leads the way. It points toward the pole it's heading toward, acting like the front of the chromosome. The rest of the chromosome, those arms, trails behind. If you watch under a microscope, the chromosomes look like they're being pulled by their centers, with the arms dragging. The spindle fibers pull the chromosomes by shortening themselves, reeling the chromosomes in. It's like fishing. The spindle fiber is the line, and as it shortens, it reels the chromosome toward the pole. Telophase is the final stage of karyokinesis, the final stage of nuclear division. By now, the chromosomes have traveled to their respective poles. They're at opposite ends of the cell. Once they arrive, something happens. They decondense, they loosen up. They're no longer these tight, compact, visible structures. They start to spread out and become less distinct. The individual chromosomes are no longer visible as separate entities. Instead, you just see masses of chromatin material, loosely organized DNA, collecting at each pole. But now the really remarkable part happens. A nuclear envelope starts reforming around each cluster of chromosomes. Not one nuclear envelope, two. Two separate nuclei are forming. It's like the cell is reconstructing the nucleus, but now there are two of them. At the same time, the nucleolus, that structure in the nucleus that makes ribosomes, reforms. The Golgi complex that disappeared, it reforms. The endoplasmic reticulum that was invisible, It rebuilds. The cell is basically putting itself back together, but now in duplicate. Everything that disappeared during prophase is coming back, and that sets the stage for cytokinesis, the physical division of the cytoplasm. After karyokinesis comes cytokinesis. Cytokinesis is the physical division of the cytoplasm. When cytokinesis is complete, you have two completely separate cells instead of one. In animal cells, cytokinesis happens in a really specific way. A furrow, an indentation, appears in the plasma membrane. It's like the cell is pinching itself. The pinch gradually deepens, cutting inward toward the center of the cell. The furrow continues to deepen, cutting deeper and deeper. Eventually, it reaches the very center of the cell and joins up completely. The cell is now pinched completely in two. The cytoplasm has been separated into two distinct portions. And importantly, during this division, the organelles, the mitochondria, the ribosomes, and other cellular structures are distributed between the two new cells. Each new cell gets some of these essential structures so it can survive and function on its own. Plant cells divide differently, and the reason is pretty obvious. They have a rigid cell wall. You can't pinch a rigid wall like you can pinch a flexible cell membrane. So plants evolved a different strategy. Instead of a furrow pinching inward from the sides, the new cell wall forms right in the middle of the cell. The cell wall builds from the center outward, growing like a ring, eventually meeting up with the existing cell walls on the sides. The science has been clear for a long time, but the evidence from this past decade is definitive. This period from 2015 to 2025 will be remembered as the decade the consequences of our inaction became undeniable. That temporary breach of 1.5 degrees served as a final, unambiguous warning. The scientific challenge now is to monitor these accelerating changes, but the human challenge is to finally close those gaps between promises and performance before those tipping points are crossed for good. Why does mitosis matter? Well, for one thing, it's how you grew. The growth of multicellular organisms is entirely due to mitosis. You started as one cell. That cell divided, and divided, and divided trillions of times. That's mitosis at work. Mitosis produces daughter cells that are genetically identical to each other into the parent cell. Each daughter cell is an exact genetic copy. That genetic fidelity is crucial. If daughter cells had different genetic information, an organism would be falling apart from the inside. Instead, through mitosis, you maintain consistency throughout your body. Here's something you might not think about. When a cell grows, the nucleus doesn't grow at the same rate as the cytoplasm. The ratio between nucleus and cytoplasm gets disrupted. That's actually a problem. The nucleus controls the cytoplasm. It sends out signals, provides instructions. If there's too much cytoplasm relative to nucleus, the cell doesn't function well. So cell division solves this problem. By dividing, the cell creates two smaller cells, each with a proper nucleus to cytoplasm ratio. It's a homeostatic mechanism, a way of maintaining balance. And then there's cell repair. Your body is constantly replacing worn-out, damaged, or aged cells. The cells on the outer layer of your skin being replaced constantly through mitosis. The cells lining your gut being replaced frequently. Your blood cells, red blood cells, white blood cells, platelets, all being produced constantly through mitosis. Every second of every day, millions of cells in your body are dividing to replace cells that have been lost. That's mitosis at work, maintaining tissue health and homeostasis. For plants, there's a special significance. Plants have regions called meristematic tissues. The apical meristem is found at the tips of growing shoots and roots. The lateral cambium is found along the stems and roots. In these meristematic tissues, cells are constantly dividing by mitosis. That's why plants can keep growing throughout their entire life. A tree can keep adding new branches, new roots, new height. That's meristematic mitosis at work. Now we're going to shift gears. We've been talking about mitosis, which produces identical daughter cells for growth and repair, but sexual reproduction works differently. Sexual reproduction involves the fusion of two gametes. Each gamete, whether it's a sperm cell or egg cell or pollen or ovule, carries a complete haploid set of chromosomes. That means half the chromosome number compared to the parent. These gametes are produced from specialized diploid cells through a very different kind of cell division. That division is called meiosis. Meiosis is a specialized kind of cell division. It's called a reduction division because it reduces the chromosome number by half. It takes a diploid cell, a cell with the full chromosome number, and produces haploid cells with half that number. These haploid cells are gametes. They're what fuse together in sexual reproduction. When two gametes fuse during fertilization, the chromosome number is restored to the diploid number. So we have this beautiful alternation. Meiosis reduces the number, creating the haploid phase. Fertilization restores it, creating the diploid phase. This cycle continues through generations. Here are the key features of meiosis. First, meiosis involves two sequential divisions. That's meiosis I and meiosis II happening back to back. But here's the critical part. There's only one DNA replication cycle, one, not two. This is different from the cell cycle we've been discussing with mitosis, where you have S phase replicating DNA once, then M phase dividing once. With meiosis, you have DNA replication once, but you have two division phases. That's how you go from diploid to haploid. Meiosis I is initiated after the parental chromosomes have been replicated during S phase. So each chromosome consists of two sister chromatids, exactly like in the cell cycle leading to mitosis. The starting configuration is the same. Each chromosome is made of two identical sister chromatids joined at the centromere. But from this point on, what happens in meiosis is very different from mitosis. The huge difference during meiosis, homologous chromosomes pair up. This process is called synapsis. Homologous chromosomes are chromosomes that carry the same genes in the same locations. Like your chromosome number 1 from mom and chromosome number 1 from dad, they're homologous. During synapsis, these homologous chromosomes pair up closely, and when they do, something remarkable happens. They exchange genetic material. We call this crossing over or recombination. The exchange happens between non-sister chromatids, between chromatids from different chromosomes in the homologous pair. This exchange creates new combinations of genes. And here's the final result. 4 haploid cells are formed by the end of meiosis II. Haploid means half the chromosome number. One diploid parent cell becomes 4 haploid cells. But here's the remarkable part. Due to crossing over that happened earlier, and due to the way homologous chromosomes are randomly distributed, each of these 4 cells is genetically unique. They're not identical like the daughter cells from mitosis. They're different. That genetic variation is incredibly important. Meiosis breaks down into 2 sets of stages. Meiosis I has prophase I, metaphase I, anaphase I, and telophase I. Meiosis II has prophase II, metaphase II, anaphase II, and telophase II. Now here's something interesting. Meiosis II actually resembles mitosis quite a bit structurally. The stages look similar, but the starting material is different. Meiosis II starts with haploid cells, cells that already have half the chromosome number. After meiosis II, you still have haploid cells, but now each chromosome has been separated from its sister chromatid. You end up with 4 genetically unique haploid cells. Think of it this way. Meiosis I is the reduction division. It separates homologous chromosomes from each other. You start with diploid, you end with haploid, after meiosis I. Meiosis II is the equational division. It's similar to mitosis in that it maintains the same ploidy level. It separates sister chromatids from each other. So the 2 divisions serve complementary purposes. Meiosis I does the heavy lifting of cutting the chromosome number in half. Meiosis II finishes the job by separating sister chromatids. Now we get into the details of meiosis. Prophase I is the most complex stage of the entire meiotic process. It's much longer and more complicated than the prophase we saw in mitosis. To understand what happens, we need to break prophase I into 5 substages. These substages are leptotene, zygotene, pachytene, diploidine, and diakinesis. Each substage has very specific chromosomal behaviors and events. This extended duration is necessary because so much important activity is happening, especially the pairing of homologous chromosomes and the exchange of genetic material. Let's go through each substage. Leptotene is the first one. During leptotene, the chromosomes become gradually visible under a light microscope. The compaction of the chromosomal material starts, and it continues throughout leptoteen. Chromatin that was loosely organized is becoming more compact, more visible, but it's a gradual process. This is the first substage, and it's relatively short-lived. Zygotane is the second substage. This is where things get really interesting. During zygotane, chromosomes start pairing together. This process of pairing is called synapsis. Homologous chromosomes, the ones from mom and dad that carry the same genes, they line up alongside each other. Paired chromosomes are called homologous chromosomes. They're organized in a very specific way, parallel to each other. During synapsis, something remarkable happens at the molecular level. Electron micrographs show that a complex structure forms called the synaptonemal complex. This is a sophisticated protein structure that literally holds the homologous chromosomes in alignment. It's like a molecular zipper holding two chromosomes together. The synaptonemal complex provides the structural framework that allows the homologous chromosomes to stay paired and eventually exchange genetic material. When two homologous chromosomes have synapsed paired up, we call this pair a bivalent. It's also called a tetrad. Both terms mean the same thing. The name tetrad makes sense when you realize that since each chromosome has already been replicated into sister chromatids, the pair of synapsed homologous chromosomes consists of 4 chromatids total. Hence tetrad 4. 4 chromatids all held together by the synaptonemal complex. This is a really key structure to understand because this is where crossing over is going to happen. Pacatine is the third substage, and it's the longest one. During pacatine, the 4 chromatids of each bivalent become very distinct and clearly visible. They appear as tetrads. And here's where it gets crucial: recombination nodules appear. These are the sites where crossing over will occur. You can actually see them under an electron microscope, little bumps or nodes along the paired chromosomes. Cross-over is the exchange of genetic material between homologous chromosomes. But specifically, it happens between non-sister chromatids. So if you have a bivalent with 4 chromatids, the exchange happens between chromatids from different chromosomes. The enzyme that catalyzes this crossing over is called recombinase. It cuts the DNA strands and swaps segments between the homologous chromosomes. This is how genetic variation gets created. Genes from mom get mixed with genes from dad. Crossing over leads to recombination of genetic material. So now you have one chromosome with a combination of genes from mom and dad. The 2 homologous chromosomes remain linked at the sites where crossing over occurred. These links hold them together. Recombination is fully completed by the end of pacatine. So by the time pacatine ends, you have chromosomes with new, recombined genetic information, gene combinations that didn't exist in either parent. Diplosin is the 4th substage. At the beginning of diplosin, the synaptonemal complex, that molecular zipper holding the homologous chromosomes together, starts to dissolve. As it breaks down, the chromosomes tend to separate from each other. But they don't separate completely. They remain linked at the sites where crossing over occurred. Those sites are still holding the chromosomes together. These linkage points have a distinctive X shape, and we call them chiasmata. The name comes from the Greek letter chi, which looks like an X. Under a microscope, these X-shaped structures are clearly visible where the chromosomes are still connected. The chiasmata are holding the recombined homologous chromosomes together. Interestingly, diplotin duration varies widely. In some cells, particularly the egg cells in female animals, diplotin can last for months or even years. The egg cell essentially pauses in diplotin until it's needed for reproduction. Diakinesis is the 5th and final substage of prophase 1. During diakinesis, something called terminalization of chiasmata occurs. The X-shaped chiasmata, the crossing over sites, move toward the ends of the chromosomes. It's like they're sliding along the chromosome toward the telomeres. The chromosomes become fully condensed, reaching maximum compaction. They're as tightly packed as they're going to get. At the same time, the meiotic spindle is assembling. The asters are forming. Everything is preparing for the separation of homologous chromosomes. By the end of helichromatase, we see familiar changes. The nucleolus disappears. The nuclear envelope breaks down. The spindle apparatus is ready. The helochromate represents the transition into metaphase prophamately. It's the completion of prophamat, that really complex 5-stage process where so much genetic shuffling has occurred. Homologous chromosomes have paired, synapsed, undergone recombination, and are now held together by chiasmata at the sites of crossing over. They're ready to separate. Metaphase I bivalent chromosomes. Remember, these are paired homologous chromosomes. They line up on the equatorial plate at the center of the cell. Microtubules from opposite poles of the spindle attach to the kinetochores. Each bivalent is being pulled from both sides, balanced at the equator. One interesting thing, a bivalent can orient either way. Homolog one from mom can be on the left or the right. There's no predetermined direction. This random orientation is actually really important for genetic variation. Anaphase figure. This is where the reduction happens. The homologous chromosomes separate from each other and move toward opposite poles. But notice, the sister chromatids do not separate. Each chromosome still consists of 2 sister chromatids attached at the centromere. So you're pulling apart homologous pairs, but the sister chromatids stay together. This is different from anaphase in mitosis. The result, each pole now has half the chromosome number of the original cell. The reduction has occurred. Telophase I, the nuclear membrane and nucleolus reform at each pole. Cytokinesis follows, and the cytoplasm divides. You now have 2 cells, called a dyad. Each cell is haploid. It has half the original chromosome number. But remember, each chromosome still consists of 2 sister chromatids. Now comes a brief pause called interkinesis. This is the gap between meiosis first and meiosis II. But here's what's critical. NO-DNA replication occurs during interkinesis. This is the key difference from the S phase between G2 and M phase in the regular cell cycle. There's no S phase, no DNA synthesis. Prophase II is much simpler than prophase I. In fact, meiosis II resembles mitosis quite a bit. Meiosis II starts right after the cytokinesis from meiosis I, usually before the chromosomes have even fully elongated. The nuclear envelope disappears. The chromosomes condense, they become compact. There are no 5 substages like in prophase I. It's straightforward and quick. Metaphase II. The chromosomes line up at the equatorial plate, the center of the cell. Each chromosome still has its 2 sister chromatids attached at the centromere. Spindle fibers attach to the kinetochores of the sister chromatids. One chromatid is being pulled from one side, its sister chromatid from the other side. It looks very similar to metaphase in mitosis. In anaphase II, the centromere is split. The sister chromatids separate from each other. Now they become individual chromosomes. These chromosomes move toward opposite poles of the cell. The spindle fibers shorten, reeling in the chromosomes. It's very similar to anaphase in mitosis, but remember, you're starting with half the chromosome number because this is a haploid cell. Telophase to come chromosomes reach their respective poles. Nuclear envelopes reform around each set. Cytokinesis follows, the cytoplasm divides, and now you have 4 haploid cells, 4 distinct genetically unique gametes. Each cell has 1 copy of each chromosome, that's the haploid condition. Each cell has undergone genetic reshuffling through crossing over. Each cell is distinct. These are the cells that will eventually become gamete, sperm, egg, pollen, whatever the organism produces. Here's something really interesting about meiosis. It's paradoxical. Meiosis reduces the chromosome number by half, yet it conserves the specific chromosome number of each species across generations. How? Through fertilization. 2 gametes fuse, and the chromosome number is restored. A human gamete has 23 chromosomes. When 2 human gametes fuse, you get 46 chromosomes again. The human chromosome number is maintained, generation after generation. But here's what's even more important. Meiosis increases genetic variability in the population. This happens through 2 major mechanisms. First, crossing over. When homologous chromosomes exchange genetic material, they create new combinations of genes. Second, independent assortment. When the homologous chromosomes randomly line up in metaphase I, they can orient either way. Different combinations of maternal and paternal chromosomes end up in each gamete. The result, tremendous genetic variation. And why does genetic variation matter? Because it's the raw material for evolution. Variations are very important for the process of evolution. In a genetically diverse population, when environmental conditions change, there's a better chance that some individuals will have genes suited to those new conditions. They'll survive and reproduce. Their genes will spread through the population. Evolution happens, species adapt. Genetic variation isn't just interesting, it's essential for life itself. Let's step back and look at the big picture. According to cell theory, cells arise from pre-existing cells. The process by which this happens is cell division. Any sexually reproducing organism, that includes humans, starts its life cycle from a single cell, a zygote. But cell division doesn't stop at adulthood. It continues throughout your life. Every second, millions of cells are dividing. That's how you grew from one cell into the roughly 37 trillion cells that make up your body. That's how you maintain yourself. The cell cycle is divided into interphase and M phase. Interphase takes up more than 95% of the time. M phase, the actual division, just 1 hour. Interphase has 3 subphases. G1 where cells grow, S where DNA gets replicated, and G2 where more growth happens. M phase includes both the nuclear division, the 4 stages of mitosis, and the cytoplasmic division, cytokinesis. The 4 stages of mitosis are prophase, metaphase, anaphase, and telophase. Mitosis produces 2 identical daughter cells. Each daughter cell is a genetic copy of the parent cell. Each has the same chromosome number as the parent. That's why it's called equational division. Mitosis is what enables growth. It's what enables tissue maintenance and repair. Your epidermis cells are replaced. Your gut lining cells are replaced. Your blood cells are produced, all through mitosis. In contrast to mitosis is meiosis. Meiosis occurs in cells that are destined to become gametes. It's called a reduction division because it reduces the chromosome number by half. You have 2 divisions, meiosis I and meiosis II, but only 1 DNA replication cycle. The result is 4 haploid cells, each with half the original chromosome number. These become your gametes, your sperm, eggs, pollen, whatever reproductive cells your organism makes. Here's what makes meiosis different. It's not just about reducing chromosome numbers, it's about creating genetic diversity. Crossing over exchanges genes between homologous chromosomes. Independent assortment randomly distributes chromosomes. The result is genetic variation, and that variation is absolutely essential. It's the raw material for evolution. It allows populations to adapt. It gives species resilience. Through meiosis and fertilization, life maintains a balance. Each generation gets a reshuffled deck of genetic cards. Some genes from mom, some from dad, recombined in new ways. That's how we get siblings who look different. That's how populations evolve. That's how life adapts.
Meet Iyal, your AI study companion. Transform PDFs into smart study guides, practice tests, and flashcards for UPSC, NEET, banking, and any competitive exam.
Master your exam preparation with Iyal.